as well as proteins inside this membrane. We'll focus on the lipids first. So we're going to lecture when the TEM was discovered, the transmission electron micrograph was discovered. They did lots and lots of sectionings to look at many different parts of the cells. And they saw that if you have two adjacent cells next to each other, you can see these structures that look like railroad tracks. And it turns out to be the plasma membrane of one cell compared to the other cell. Each membrane bilayer is about 5 nanometers thick, which is about 50 atoms across. And you don't have to memorize these dimensions, but they will come back when we talk about membrane structures. Another technique that was used to study membranes is called the freeze fracture EM. And in this case, a cell is frozen and then it's cracked with the knife blade. And what happens is usually the phospholipid bilayer will split into two layers. And so you can actually focus in on, you can take an EM of the bottom layer and compare it to the top layer. So on the picture, the EM picture on the bottom right, what you'll see is that on the bilayer, they can see lots and lots of pits and lots of bumps. And this explained that proteins were active.
So we're going to explain about membrane lipids, and the most predominant ones are the phospholipids we've already talked about. So again, they are amphipathic. They have a phosphate head that is hydrophilic, and then the hydrophobic fatty acids, and they form a bilayer. Then we also have glycolipids, and glycolipids, as its name implied, glyco stands for carbohydrates, so these are lipids that are covalently linked to a carbohydrate. And the carbohydrates, if you notice on this picture, are always on one side of the membrane only. So that would be on the external side of the cell. They would not be found in the cytoplasm. Then the third type of lipids are the cholesterol, and the cholesterol we already mentioned are amphipathic, and they have a OH hydroxyl group that is the hydrophilic side. So all of them together shows that the membrane is actually asymmetrical. You can see that carbohydrates on the external side only, and on the cytoplasmic side, they do not have them. And also, they do not look exactly the same on top versus bottom. You can have different makeup, different types of phospholipids on one side versus the other side of the membrane. This picture explains how phospholipids can actually move. So they do not flip-flop back and forth from one monolayer to the other monolayer, but they are able to rotate or flex pretty easily, and they also move by lateral diffusion. So this is an experiment that shows how the phospholipids and actually proteins do lateral diffusion. So a mouse cell was labeled with an orange marker, and then the outside of a human cell was marked with blue markers. And then the cells are brought together and fused, and after a little while, you'll see that the orange and blue were all intermixed everywhere, and that shows that lateral diffusion had to have taken place. A technique that was used to try to determine how many different types of lipids are found in the membrane is called the thin layer chromatography. So this technique is very similar to the paper chromatography that we had done in lab. And basically you have a plate. It's can be a plastic sheet or a glass plate coated with either silica or cellulose. And then you would take your mixture of lipids that were extracted from your membranes or whatever sample, and you put that onto the origin at the bottom. And you put the whole glass plate or plastic sheet into a solvent, And the solvent usually is an organic solvent, a hydrophobic solvent. And then you wait for the solvent to move through capillary action up the plate. And if you look at the chromatogram here, you can see cholesterol, PE, PC, PS. Those are all different types of phospholipids, and you would need to make interpretation. So if this was a very non-hydrophobic solvent, then you would say cholesterol is the most non-polar molecule, and then PE, and then PC, and then PS. If the solvent had been more polar, and this is the spots that you get, then you would say cholesterol is the most polar. This type of experiment was done in in many different membranes, and so here you see the data from a rat liver, potato, bacteria, a human nerve, a spinach chloroplast, and then rat mitochondria. And after doing the TLCs to decide what types of phospholipids there were, then the next thing was to quantitate them. And so this is what the graphs are showing. The interpretation of these is that every membrane has a different composition, and it's a composition of phospholipids, and it's likely that every membrane has a different function, and so the composition has to reflect that function. This picture shows a different concept, and that is we would like to know when some of the phospholipids actually melt or go from a solid state to a liquid state. So an example of this is in your kitchen. You have butter that is from animal fat and saturated fatty acids, and then you have corn oil, which is unsaturated fatty acids. And there's a big difference. So typically, if you have an unsaturated fatty acid, like the oleate that's shown on the left, you'll get a melting temperature that's low. So in this case, 16 degrees Celsius. And so that means below 16 degrees, oleate was a solid. And above 16 degrees, oleate is a liquid. And at room temperature, which is about 22 degrees, then oleate is a liquid, which makes sense because that's what our unsaturated corn oil seems to look like. Then the opposite is stearate on our right, and stearate is a saturated fatty acid with straight chains, and it is a solid until it hits about 70 degrees, and then it melts. So that sounds right. That matches our butter, which is a saturated fatty acid. It's going to be a solid at room temperature at 22 degrees. Carrying that one step further, or two steps further, we actually have this orange graph, and in this experiment, they took fatty acids of different carbon lengths and asked what is the melting temperature change. And the general trend is as you increase the carbon atom's length, then you also increase the melting temperature. On the right graph, which is red, you see that as you increase the number of double bonds in a fatty acid, the melting temperature will decrease. So that's an inverse relationship. It makes sense if you look at the structure of the fatty acids. The top left shows you saturated fatty acids that are straight chain, and so it's much easier to pack these straight chains together in the membrane. Whereas in the bottom you have unsaturated fatty acids that the bent chains, bent chains, then it's much harder to pack them together. And so the unsaturated fatty acids tend to retain more fluidity. For cholesterol, you have an alpha-pathing molecule, and so the OH or hydroxyl group is going to make hydrogen bonds with the fatty acid hydrophilic heads, and then the 4-ring structures are going to form hydrophobic interactions with the fatty acids. So putting all this together, I have an application question that I'll give you ahead of this exam. And you are supposed to think about a creature that's living at 30 degrees. What happens if you put it into a cold temperature and somehow it survives? We would like to know what type of changes it had to make in the cell membrane structure in order to survive. So I want you to think about the number of carbons, the number of double bonds, and the number of cholesterol that should be either added or taken away. And the converse of that would be to take the cell from a nice temperature and go very, very high, and what would the changes